In the previous video, we looked at the first step of glycolysis. That's the conversion of glucose to glucose 6-phosphate. And we talked about how there's two different enzymes that can catalyze this reaction, either glucokinase or hexokinase. Hexokinase has a little bit more flexible substrate specificity, whereas glucokinase can really only react with 6-carbon hexoses. So that's aldose sugars like glucose or mannose. We also established that the requirement for both of these enzymes is to have an active site where both ATP and glucose can bind and can position the two molecules so the 6th carbon of glucose is oriented towards the 3rd phosphate of ATP. In this video, we're going to explore these structures in a little bit more detail. We'll compare the two enzymes, glucokinase and hexokinase, talk about the similarities and differences, and then we'll examine glucokinase in more detail and really kind of think about how this enzyme functions. And we'll end with an important detail of it that makes it a really nice target when designing drugs to battle the disease diabetes. So let's go ahead and start off by examining the overall structure of both of these enzymes. On the left here, we see the enzyme hexokinase, and on the right, we see the enzyme glucokinase. And you can really see some major similarities. Both of these enzymes have the same general architecture where it looks kind of like a clamp that wraps around the active site. And this should make sense based on what we've learned about how enzymes work. A lot of the enzyme is meant to be a scaffold that creates an active site that can bind to our substrates. And that's exactly what these two enzymes are doing. Okay, so let's go ahead now and examine these active sites in a little bit more detail. Let's start with glucokinase, which we've seen in the previous video. As we saw before, the enzyme creates a very attractive binding site for glucose. It creates many hydrogen bonds between the enzyme and glucose, which creates a lot of substrate specificity and a high affinity interaction. If we do the same thing and look at the active site of hexokinase, we see a very similar situation. The enzyme presents a cavity that has many hydrogen bonding opportunities for glucose. But as it turns out, this enzyme can accommodate both aldohexoses and ketohexoses. So hexokinase can actually convert fructose to fructose 6-phosphate, as well as converting glucose to glucose 6-phosphate. Okay, so let's go ahead and turn our attention now to glucokinase because more structural information is known about this enzyme. So one really interesting thing to note is that for both of these enzymes, you can pick out two different distinct domains. The two domains are on the top and the bottom of the active site respectively. And if we rotate this around, you can very clearly see here that this top domain, covered in purple, kind of forms a lid that closes on top of ATP and glucose. And in fact, completely shields glucose from the solvent under these conditions. We can confirm that by showing an overlay of what the surface of the protein looks like. And that's what I'm doing here with this orange surface rendering. If we rotate the molecule using this surface rendering, what we can see is that the glucose is truly shielded from the solvent, from the outside of the protein on all sides. However, as we finish this rotation, you can start to see how ATP is actually exposed. We see the base part of ATP, present in white and blue, situated in a nice groove that the enzyme creates for it. This allows the 3 phosphates to be pointed in towards the glucose molecule, which of course we know we need for this enzyme to function properly. The important thing to take home is that the ATP is exposed to the surface and can freely diffuse in and out of the active site, while the glucose is buried inside the protein throughout the entire structure, and it is not exposed to the solvent. The reason this is important to note is that it tells us that this protein must be able to adopt at least one other structure, because otherwise, how would the glucose ever get into the active site? Fortunately, scientists have been able to determine the structure of this enzyme in a different conformation. So what I'm showing here is the other conformation, which may or may not be useful on its own, but it's definitely useful if we superimpose these two structures on top of each other, which is what I'm doing here. And what you can clearly see is that there are some regions of the protein that are essentially identical, especially in that lower bottom domain. We see perfect overlap between both of these structures. But on that top domain, which we can describe as kind of a lid over the active site, we can see some very clear differences. In fact, we see that in this other configuration, the lid seems to have been lifted. This is certainly reinforced when looking at the surface image of the enzyme. In the yellow conformation, we see a more open active site where the lid is not closed, compared to the closed configuration in orange where that lid has been closed over the active site of the enzyme. And just to reinforce this idea, I'm now showing the surface rendering in a little bit different conformation where we can see the ATP. And remember that ATP is leading into the glucose binding site. In one conformation, the orange, we see the lid closed over the active site, whereas in the yellow conformation, we see that lid has been opened up. So what this begins to suggest to us is that there must be a mechanism that the enzyme can use to convert between these two different structural conformations. And if we think about the two domains and think about this idea that there can be an opening and a closing of the clamp, then what that begins to suggest is that perhaps there's a hinge region. So let's look at the two structures overlaid again. We can propose that at the back of the enzyme is where it's most likely that there can be a hinge that opens and closes the clamp. So if we zoom in and look at that region, what starts to stick out is there's this one alpha helix that has a very clear and distinct difference in structure between these two conformations. So perhaps the origin of this structural change, of this hinge opening and closing, lies with a reorganization of that alpha helix. So when scientists noticed this difference, they were able to begin thinking about how we could design organic molecules that could interact with one conformation, the active conformation of the enzyme, and would prevent the enzyme from being in a conformation where it's not active. What the scientists ultimately came up with was a series of molecules that can bind at this site and can force the alpha helix to be in the conformation that corresponds to the active conformation of the enzyme. If we look closely, you can see how this molecule, when it's bound, binds too closely to the alpha helix in the yellow conformation, so it forces it into the other conformation, and that's the active conformation. So this is a great example of how we can design a drug that will force the enzyme to be in an active conformation and not in the inactive conformation. And ultimately what this does for us is it helps us ensure that this enzyme, glucokinase, is functioning as well as it possibly can. And this was a really important idea to help scientists combat diabetes, which we'll talk more about a little bit later in class.